Hello guys, today's topic is nutritional requirements for plant tissue cultures. So first of all, we will discuss plant tissue culture. What is plant tissue culture? Tissue culture is in vitro cultivation of plant cell or tissue under aseptic and controlled environment conditions in liquid or on semi-solid well-defined nutrient media for the production of primary and secondary metabolites और टू रीजनरेट प्लांट्स। टिश्यू कल्चर एक इन विट्रो कल्टीवेशन है जिसमें प्लांट सेल और टिश्यू को एक एसेप्टिक और कंट्रोल्ड एनवायरनमेंट में लिक्विड और सेमी सॉलिड न्यूट्रिएंट मीडियम में रखा जाता है, जिसमें कि उस प्लांट टिश्यू की ग्रोथ करने के लिए सारे न्यूट्रिएंट्स मौजूद होते हैं, प्रेजेंट होते हैं। दिस इज कॉल्ड प्लांट टिश्यू कल्चर। Optimal growth and morphogenesis of tissue may vary for different plants according to their nutritional requirements. Moreover, tissue from different parts of plants may also have different requirements for satisfactory growth. So, tissue culture media were first developed from nutrient solutions used for culturing whole plants. Example: root culture media of white and callus culture media of gotherate. बेसिक मीडिया दैट आर फ्रीक्वेंटली यूज इंक्लूड, जो बेसिक मीडिया हैं जो यूज किए जाते हैं प्लांट टिश्यू कल्चर में, वो हैं मॉर्सिंग एंड स्कूग मीडियम, लेंसमियर एंड स्कूग मीडियम, गैम्बोर्ग मीडियम एंड निश्च एन मीडिया। सो न्यूट्रिशनल रिक्वायरमेंट्स फॉर प्लांट टिश्यू कल्चर media composition. Plant tissue culture media should generally contain some or all of the following components: macronutrients, micronutrients, vitamins, amino acids or nitrogen supplements, sources of carbon, undefined organic supplements, growth regulators and solidifying agents. So macronutrients. What is macronutrients? The essential elements in plant cell or tissue culture media include, besides carbon, hydrogen and oxygen, macro elements nitrogen, phosphorus, potassium, calcium, magnesium and sulfur for satisfactory growth and morphogenesis. Culture media should contain at least 25 to 60 mm of inorganic nitrogen for satisfactory plant cell growth. पोटेशियम इज रिक्वायर्ड फॉर सेल ग्रोथ और मोस्ट प्लांट स्पीसीज। जो कि कल्चर मीडिया है उसमें एट लीस्ट 25 to 60 mm इनऑर्गेनिक नाइट्रोजन होना चाहिए फॉर किसके लिए? सेटिस्फैक्टरी ग्रोथ ऑफ प्लांट्स के लिए। पोटेशियम इज रिक्वायर्ड फॉर सेल ग्रोथ ऑफ मोस्ट प्लांट स्पीसीज। मोस्ट मीडिया कंटेन पोटेशियम इन द फॉर्म ऑफ नाइट्रेट, क्लोराइड, साल्ट एट कंसंट्रेशन रेंजिंग बिटवीन 20 and 30 mm. The optimum concentration of potassium, magnesium, sulfur and calcium range from 1 to 3 mm if other requirements for cell growth are provided. Micronutrients. The essential micronutrients, also known as minor elements for plant cell and tissue growth, include iron, magnesium, zinc, boron, copper and molybdenum. Iron is usually the most critical of all the micronutrients. The element is used as either citrate or tartrate salts in culture media. However, there exist some problems with these compounds for their difficulty to dissolve and precipitate after media preparations. There has been trials to solve these problems by using ethylene diamine tetraacetate acid EDTA, carbon and energy sources for plant tissue culture. In plant cell culture media, besides the sucrose frequently used as a carbon source at a concentration of 2 to 5, other carbohydrates are also used. These include lactose, galactose, maltose and starch, and they were reported to be less effective than either sucrose or glucose. The latter was similarly more effective than fructose, considering that glucose is utilized by the cell in the beginning, followed by fructose. Vitamins and vitamins and MYO-INOSITOL. Some plants are able to synthesize the essential requirements of vitamins for their growth. Some vitamins are required for normal growth and development of plants. They are required by plants as a catalyst in various metabolic processes. They may act as limiting factors for cell growth and differentiation when plant cell and tissues are grown in vitro. The vitamins most used in the cell and tissue culture media include thiamine, B1, nicotinic acid, and pyridoxine. Thiamine is necessary required by all cells for growth. Thiamine is used at a concentration ranging from 0.1 to 10 mg. Nicotinic acid and pyridoxine, however, not essential for cell growth of many species. They are often added to culture media. Glycine. This is also amino acid. Glycine is the most common amino acid used in different culture media. It is not essentially but nitrogen containing amino acid enhance growth and plant. Undefined organic supplements. Undefined organic supplements are some media where supplement with natural substance or extract such as protein, hydrolysate, coconut milk, yeast extract, malt extract, ground banana, orange juice and tomato juice to test their effect on growth enhancement. Plant growth regulators are important in plant tissue culture since they play a vital role in stem elongation, tropism, and apical dominance. They are generally classified into the following groups. First one is auxins, cytokinins, gibberellins, and abscisic acid. Auxin. Auxin essentially for cell division, cell elongation, cell differentiation, organogenesis, and embryogenesis, callus formation. Natural form auxin are IA, IBA, PAA. Synthetic form of auxin are Na2,4-D. Cytokinins. Cytokinins promote cell division, suit proliferation, and influence the cell cycle, embryogenesis, and inhibit root formation. Synthetic form is 2IP, which is most active. Cytokinins natural form are BAP and kinetin. Gibberellins. It promotes stem elongation, valve formation, and embryo maturation, but can inhibit callus growth and root induction. GA3 is most common gibberellins. Abscisic acid. It inhibits shoot growth and germination of embryo. It is thermostable but light sensitive होता है. Solidifying agents. Hardness of the culture media greatly influence the growth of cultured tissue. There are a number of gelling agents such as agar, agarose, and gelling gum. pH of tissue culture media. pH is adjusted between 5 to 5.8 before gelling and sterilization with the help of dilute NaOH, KOH or HCl. pH below 5 will not properly. pH above 6 may be too hard. So thank you all of you. Please do subscribe my channel, share, like. Thank you.